for being here. So my name's Erik Wright. I'm from the University of Pittsburgh in the Department of Biomedical Informatics. And today I'll be talking about predicting gene functional associations from coevolutionary signals with our program EvoWeaver. I'd just like to point out before I begin that a large fraction of this work was done by Aidan Lakshman, who's in the audience and has a poster that he'll be presenting tomorrow. And he actually won the poster at Recom for this. So it's pretty cool work and I'm excited to be here. I'd like to start with something very fundamental, and that is a random protein sequence from a random metagenome. And I promise you, this is the first random protein sequence. So we're gonna get some results from it. We'll see how it goes. From the first random metagenome I selected completely randomly. And I like that when I take a step back and think about what we offer as scientists, I think one of our primary contributions is understanding more about life. And so I want to think about what we as scientists can add as value to this random protein sequence. And I think you might say, well, scientists can run experiments, and that might be the first thing that you think of. And that is a lot of how we've learned what we've learned about life. And I'm sure all of you know that's not so easy. It takes a lot of effort and time and money. But I also think that another big issue with experiments, because I'm an experimentalist myself, is that you don't know what to do lots of times. It's unclear. So I gave you a random protein. Where do I go from here? And maybe you've got some ideas, but it would be helpful to narrow that down substantially. So, but we're bioinformaticians in this room, right? So we can do better. So you might say, well, you can fold it. And I bet a lot of you would think of that. And that's a great suggestion. So here I folded it. There it is. And for more than half our proteins, we have these beautiful structures, right? And AlphaFold thinks this is probably the right structure for almost all of it. So that's wonderful. And what do I do with it now? For some proteins, you can look at them and learn stuff like transmembrane proteins. But for this particular protein, I mean, personally, when I look at this, I don't know what to do with it. But you might say, well, we can do better. We can search for it. And that's a great next step. So here's fold-seq output. This is the top hit. And in this case, we get a hit that fold-seq thinks is wonderful, right? It's a pretty decent e-value. Probability of the hit is 100%. Not the best TM score, but you can see it's a reasonable fit structurally. So what did we learn from searching for it? Well, we learned who its nearest neighbor was in this UniProt database. We learned that it's potentially a sulfotransferase. And if you go way down the list of proteins that are hits, they're all sulfotransferases. And so good, we're done, right? But the issue is, well, there's a lot of different sulfotransferases. And I think this is pretty representative in that you get back a result, but that result is not particularly informative. If you're a biologist, you want to go deeper. So the next thing that I'm sure all of you are thinking of in this room is, well, you can network it, right? That's what you were thinking. I'm hoping at the end of this talk, that is what you think. So, and some folks would do that with string. And that's a great idea. And in fact, it worked in this case. So you have a functionally associated potential partner. And that partner is a senescence related protein. So now we've got some idea of where to go with an experiment if we're an experimentalist. And this was helpful, I think. So potentially this is part of a senescence pathway. And that might narrow down what our experiments might be. So the question is, how well does this work? And can we make it better? If you look at where string is getting its evidence from in this particular case, it's getting most of it from databases and text mining and some from experiments. That's pretty normal for string. And in fact, if you do a breakdown of where string gets its evidence from, these are the contributions of their different 7 evidence streams to their total score, which is what you usually are working with string. And you can see more than half of string is mostly coming from text mining in other databases. So string is just a database that is derived from other databases, which makes sense, and that's okay. Another good fraction of its data is that of experimental data. And then less than a quarter of its data or its signal, is coming from what I would call coevolutionary signals. In this case, there's 3 coevolutionary signals, and the strongest is gene neighborhood. This is representative of all of string, and obviously it would be different depending on which protein you look at, where it would get its signals from, but this is the aggregate. What I'm interested in is, can we do better than this? Is it possible to make a giant improvement upon this, specifically with coevolutionary signals? Because if I'm interested in understudied proteins, and here I have this random protein from a random metagenome, I'm not going to normally have a lot of information from databases and text mining. In this particular case, because I selected a random one, we got lucky. But most of the time, if I have an understudied protein, that's not where I'm going to get my information from, right? I'm going to get it from some source of data that's independent of what we already know is our knowledge bases. So when I look at these 4 possibilities in bioinformatics, we can run an experiment that's hard. We can fold it. That's somewhat of a solved problem now, right? It works very well. We can search for it. Nowadays with PLM search and fold seek, we can do a pretty good job of that. And I think with networking it, which is, in my mind, the thing that has the most potential of anything up here, is that we can improve databases like string by adding signal to them. So much of our information in biology is encodable in networks, right? Because the cell is a network of different things that interact with each other, either physically, like a PPI, or they simply work on the same substrates. And one of our biggest networks that houses a lot of information is KEGG. And KEGG has this global path of metabolism that's beautiful, and I'm sure some of you have seen it before. It has things in it like how our cells make energy through oxidative phosphorylation. It has things like the TCA cycle. And each of these nodes here is a molecule in this network. And so we have oxaloacetate, and it gets converted to citrate, and that's done by the EDGE, which is an enzyme called citrate synthase. So many of you are probably familiar with this in the TCA cycle. I'd like to just focus on a different part of this network here. And it doesn't really matter what I pick. Anything could be used as an example of this. But here it's biosynthesis of an antibiotic as part of some organism's secondary metabolism. And I'm just going to look at 2 genes that are adjacent in this network. Because if I can predict that 2 genes are adjacent in this network, then effectively I can predict much of this network. So I'd have a lot of power if I can predict that val-C and val-K are adjacent in this network relative to all the other proteins. So let's just look at different sources of evolutionary signal that exist. Commonly researched source is patterns of presence and absence. So here you can see that val-C and val-K on the organisms in which one or the other is present, they oftentimes appear together, right? So that's of interest. That's a signal that these 2 things might be functionally associated, but it's not a very strong signal, especially in this case. Another pattern that you might look at is how they're co-located on genomes. Especially in prokaryotes, you have a lot of signal here, and eukaryotes maybe a little less. But nevertheless, there's quite a bit of signal encoded in where these genes are. Val-C and val-K tend to be near each other. They tend to be in the similar direction on similar strand. And so we call this pattern colocalization. And that's the second pattern we'll look at today. The third is that their phylogenetic trees, their gene trees for val-C and val-K, one on the left, the other on the right, are fairly similar to each other, right? The one difference in topology I'm showing is a dotted branch that happens to be very high in the phylogeny, where it's a little bit harder to resolve. It's a much shorter branch. And you can see the tangulogram shows that these 2 things are similar gene trees. So there's a little bit of signal in correlated trees. And there's also residue covariation. So this is much of the signal that we see for things that interact, like PPIs, where they actually functionally touch each other. And so we can potentially parse this signal to get more resolution in terms of our prediction of functional association. And if I want to test these signals against each other, I can take something like KEGG, and I can look at the adjacent interaction or adjacent edges, and I can say that those 2 things are functionally co-evolving with each other. They're functionally associated, and I can call that a positive, and I can call a negative something between pathways, right? And so KEGG's very big. It has on the order of 25,000 of these ortholog groups that represent an edge. It has on the order of 600 different pathways that are the colored lines here. And I can construct an ROC curve for this. So if we look at how our 4 different measures of coevolutionary signal look on this ROC curve for predicting positives versus negatives, we can see the pattern of presence absence does a pretty good job, right? We, with just a few false positives, managed to collect about half the signal. Co-localization also does a fantastic job. So this is another pattern, and that pattern might be somewhat orthogonal to the first pattern that we looked at. The third pattern does an okay job. And the fourth one, which is typical of this type of data, is the patterns based on the 2 proteins touching each other. For a data set like this, where the proteins in KEGG do not have to touch each other, doesn't necessarily do a great job. And so I have these 4 patterns. And so what we've done in EvoWeaver is we've parsed these patterns out of simply gene trees. And the advantage here is all we need are genomes. We have no prior information. We don't know what anything's called. We don't know what any of these nodes or edges are labeled. We know nothing. We have no text data. We have no databases. Here we're completely de novo. All we have is genomes. We call genes, and then we can construct gene trees. And if we do that, we can get these patterns straight out of there, and it tells us a lot about biology already. And we've spent a lot of time working on this. And so it would be a little bit oversimplifying. And I have to fit it all in this talk, but I'll just tell you there's many other signals that we look at. In fact, there's 12 total. There they are. And then what we realize is it's straightforward to just add those signals together and put them into an ensemble prediction. And you can do this with a random forest quite simply if you'd like. You get an excellent result, okay? So we're able to predict positives from negatives very well, and we're actually able to do this much better than string in the absence of existing knowledge, okay? So you might say, well, something I hear a lot in these PPI talks that I've been at the last couple of days is, well, you have lots of data issues, data leakage issues when you train. Well, none of these 4 patterns are trained. They're just scores. They come directly from the data. You're looking at tree distance, things like this. You're just simply taking RF distance. That doesn't require any training. So all of the patterns by themselves have no training. If you look at the random forest, you could say, well, that's overtrained because that does have training. That knows what a positive is and a negative is in the training set. And we've done a lot to mitigate overtraining and reduce data leakage to the maximum amount. But I can tell you that a linear regression, which should be less overtrained, does basically as well as random forest. So this signal, as you might imagine, there's lots of different orthologous predictors. You can put them together, you can get a better result. So that's what we've done in EvoWeaver. We're super excited about it because we think that this is going to, as we continue, revolutionize the way that we extract knowledge from genomes. And if we just zoom in on the part of the curve that really matters to me, I want to have a low false positive rate, right? I don't care about the entire curve. I'm more interested in the part off to the far left. And there you can see that we're still doing pretty well. If you are allowing a few false positives because maybe KEGG has some edges missing from it, then if you allow for that, then you can pick up with our ensemble predictor a fair amount of the true positives. So that's a fabulous result. And it's wonderful that it works. And so what can we do with it? Well, I'll go back to KEGG here. And I'll just say, if I predict all of the different edges here against all the other edges, and then I just simply cluster the data with a community detection algorithm like Leiden clustering, I can pull out pathways from this, which is brilliant, right? If I'm a biologist and I have a new protein, I say, which proteins am I working with? This has enough exquisite signal to it. It's able to detect false positives from true positives so well that you can pull out entire pathways from this just directly. And so there we get this pathway. It falls right out of the data. This is to create a different antibiotic. It's also part of secondary metabolism for some organisms. And this is what the pathway looks like in KEGG. So we start off with a molecule that goes through these genes, and then it creates a different molecule, which happens to be an antibiotic. And if we look at what our predictions are on this pathway, we see that we get some of the edges that we expect. We're missing other edges that we did expect. And we have quite a number of cross edges. And this seems to be the case for a lot of our work, where things are all, this entire pathway is co-evolving. So it's very difficult to say that, you know, back A is co-evolving with back B, but I want to make sure that I don't connect it to back C, right? Because those things are still co-evolving. And so you have some degree of bleed through where the information is shared across all the proteins. But the cool thing about this is you can immediately pick out these parts of the network. So when you cluster it, they fall out together. And if you're an experimentalist and you know that back A is the protein you're interested in, you want to know what it does, this could help you greatly to narrow down what proteins it might be functionally associated with and what these might do together. Especially if more information is known about something like back B, you can potentially transfer that information to back A. So this is part of EvoWeaver. EvoWeaver is one of many tools I presented on Clusterize last year in this same track. All these tools come from our lab. Some of them are published. Some of them will be published soon. EvoWeaver's publication, unfortunately, is not here in time for the conference. I was crossing my fingers, but it's not quite. So you can expect to see that soon, I hope. These are all part of the SynExtend package, which has been in Bioconductor for 4 years. Our main package, Decipher, which is the one that I maintain, has been in Bioconductor for 13 years. And you can give it a shot if you'd like. You can go ahead and download EvoWeaver, and you can try out all these different predictors on your protein of interest. All you need is the protein sequences themselves. We can create alignments. We can create trees using these other tools. And then, boom, you have the data that I showed. So it's very straightforward, very scalable, very fast, works extremely well. And we're just going to make it better and better. With that, I'd like to thank my group. Aiden Lakshman, who's here, is sitting next to me. If you'd like to contact me, please do. That's our lab website. And this work was supported by the NIAID. Thank you.
Thank you very much for the interesting talk. Any questions? Yep. I predicted you would have a question.
Okay. Thank you for the talk. Very nice. Why testing is fine, testing with cake, but I will go to a string and test a bit against the part of a string that is already from derived from databases and text, as you already explained.
Yeah, so we've compared to string. I didn't show the data here for lack of time. We've compared to string directly on KEGG and also on another data set called Quorum. And so we've tried to do our due diligence and go beyond just KEGG and see what's out there. I'd say one of the major challenges that I didn't touch on is that the underlying data that goes into this is very essential. I don't think KEGG's or ortholog groups are quite perfect. And because of that, I think we can actually do better. And that's a big thing that our team's working on, is making better underlying data in terms of ortholog groups to drive these algorithms to create better signal. And so I don't personally, I'm not married to any given database. I think we're going to end up ultimately needing to make our own data.
Great talk, thanks. So a couple of things. You took your phylogenomic data and your genomic co-location data from string or from somewhere else, because I can seem to take only network data from string, but string also has co-location, and you can also get your phylogenomics data from there.
So we're great question. So we're comparing against KEGG, and we're deriving all our information from KEGG for what I showed. We compare indirectly against string on KEGG proteins, for example, but we're not using string as our input data. We could. There's nothing to stop us from doing that. KEGG contains co-location data, so it tells you where the gene came from in the genome. It also contains the ortholog groups. And because we make our own program for constructing orthologs, which is part of SynExtend that I showed, we didn't want to have a bias in publication where people said, oh, well, you're using your own orthologs, maybe they're special orthologs. So we're trying to use other people's orthologs. I will say the underlying data, like I said, is very important, but it's also very difficult to find the amount and curation of data that we need. So there's not too many databases out there that fit our needs very well.
And my second question is, have you tried comparing your ensemble methods to other similar ensemble methods like the ones competing in CAFA? Maybe it's not possible if you're not using genome ontologies, but have you kind of looked into at least the components of, say, methods that use PLN, text mining, phylogenomics, co-location, and so on?
So no, not yet.
Okay, thanks. Yeah. Really great talk. Thank you.
Thank you.
One more question? Maybe? Yeah, there's maybe a minute.
I have many, but as I understand it, the predictions will not be species specific, will be for all the sequences in the sequence alignment, what create a problem in terms of validation, no? Because then you are validating against a given set. And that has always been a problem in the field. No? And then I have one more question before he kicked me out is, you mentioned all the other methods. So what are all the other methods that you show for the prediction? You showed we have many other methods and you didn't say which way.
Yeah, sure. So I'm so bad when you ask me two questions. So your first question, remind me, was species specific, right? Thank you. My memory is just awful. So species specific. So string is species specific, and we can do our predictions in such a way. We haven't here. We've done it based off ortholog groups. We haven't tried species-specific predictions yet, but it is possible. Regarding the alternative methods that I showed, the 12, what we tried to do is construct as orthogonal as possible signals from the data so that we can learn from them. Because if they're all correlated very strongly with each other, there's no additional information. We've spent a lot of time on that, and you can look up in SynXtend what they are if you're interested in going to that level of depth. They all fall under the 4 categories I showed, though. So we have no 5th category. They all are lumped in there. And there's, for example, I'll give you the example of correlated trees. So you can look at correlation in distances, things like mirror tree and context tree. You can also look at tree topology. And then there's many, many, many different measures of tree topology that you could try. And we've actually tried many of them. So a lot of this is implemented, and it's a tour de force that Aiden was able to do this. And he's a very, very impressive graduate student. So I'll just put that out there. There's a lot of programming.